O Grupo Onyx-Can é pioneiro quando o assunto é cannabis medicinal, transformando ciência em saúde. No Brasil, somos os únicos a trazer uma linha completa de produtos de cannabis medicinal, incluindo produtos com maior teor de THC no padrão Farma de produção. Nossa missão? Melhorar a saúde e qualidade de vida. Nossa visão? Revolucionar a forma de tratamento e o sistema de saúde do brasileiro. Nossos valores? Comprometimento com a saúde com transparência, de forma responsável para garantir segurança e bem-estar. A Onyx-Can conta com 4 pilares estratégicos que garantem a eficiência do tratamento canabinoide. São eles: educação para o médico que busca aprofundar seus conhecimentos de forma científica e para o paciente que busca informação para um tratamento seguro de cannabis medicinal. Inovação e tecnologia digital para o acesso do paciente ao tratamento médico. Farmacêutica com uma linha completa de produtos de cannabis medicinal, tendo uma linha própria de produtos, a Tegra Allyn e a Tegra Euroline. E ainda possui acordos internacionais exclusivos com a MGC Pharma e Citec para importação de produtos, incluindo pesquisas científicas e registros de produtos fitoterápicos e naturais, com tecnologia farma de ponta, e na divulgação de notícias, casos de pacientes e pesquisas, em parceria com a maior fonte de informação da medicina canabinoide brasileira, o portal de notícias Cannabis e Saúde, a maior plataforma de agendamentos do país. Conectamos você aos melhores médicos prescritores de cannabis medicinal do país, incluindo o Medical Cannabis Summit, o maior e melhor evento de cannabis medicinal do Brasil. Se o assunto é cannabis medicinal, o Grupo Onyx-Can oferece o que há de melhor e mais moderno, incluindo informação, educação, tecnologia e saúde, com transparência, segurança e responsabilidade. Tegra Pharma é a farmacêutica do Grupo Onyx-Can. Sua finalidade é distribuir, importar e comercializar uma variedade de produtos de cannabis medicinal, naturais e fitoterápicos, e com tecnologia farmacêutica capazes de suprir necessidades médicas significativas, complementando os avanços da medicina. Nossa visão: oferecer produtos para novas formas de tratamento, tendo o paciente como protagonista do seu processo de cura, saúde e bem-estar. Nossos valores: como norteamos os negócios da empresa. Nosso diferencial: aliar pesquisa com tecnologia avançada, a fim de garantir o que há de melhor na produção e comercialização de cannabis medicinal e produtos inovadores no mercado. Tudo isso em acordo com as agências reguladoras de saúde, atendendo normas e especificações de qualidade. Divididos em 2 linhas exclusivas, oferecemos produtos com diferentes concentrações de THC e CBD. A primeira, a linha Tegra Allyn, e por fim, a linha Tegra Euroline, utilizando extratos de cannabis de alta qualidade, preservando maior gama de cannabis medicinal e outros compostos importantes da planta. Contamos com parceiros especializados para oferecer o que há de melhor nos produtos de cannabis medicinal. Um deles é a MGC Pharma, uma das principais indústrias farmacêuticas internacionais no setor de cannabis. A MGC é considerada pela Bolsa de Londres uma empresa de biotecnologia e biofarmacêutica. Por isso, é uma das poucas empresas de cannabis medicinal autorizada a negociar nessa bolsa. Seu catálogo apresenta 5 produtos, de diferentes concentrações já lançados no Brasil, com exclusividade pela OnixCan. 2 deles vêm para substituir com vantagens os produtos com menos de 0,3 de THC. Outros 3 com maiores teores de THC são uma opção farmacêutica de qualidade e ainda mais barata para substituir os atuais óleos artesanais feitos no Brasil. Outro parceiro é a Citec, empresa europeia com forte investimento na pesquisa e produção científica da cannabis. Dentre as opções do seu catálogo, oferecemos 5 produtos. Com responsabilidade e comprometimento, garantimos qualidade de vida para quem necessita dos benefícios canábicos. Tegra Pharma, a farmacêutica do grupo OnixCan.
Não escolhemos a medicina, somos escolhidos por ela. Mais do que tratar pacientes, temos interesse pelo ser humano como um todo, sua família, seu trabalho, suas condições sociais. Somos tomados por uma vontade íntima de ajudar, cuidar e fazer a diferença. Investigamos sintomas, diagnosticamos e buscamos todas as formas de prevenção e cura. Nosso pacto de cuidado é construído, não só prescrito. Em um planeta adoecido, nossa missão é oferecer cuidados personalizados, holísticos, equitativos, baseados em dados, com tecnologia, biotecnologia e em todos os níveis de atenção, básicos, especializados ou de alta complexidade. Nossa inspiração vem do desejo legítimo de contribuir para uma sociedade melhor. Acreditamos no trabalho multiprofissional e interdisciplinar em uma equipe horizontal, na reflexão diária sobre a prática e no aperfeiçoamento constante. Como médicos, estudantes e educadores de medicina, devemos zelar por quem nos procura. Mas precisamos ir além, em busca daqueles que não têm acesso aos serviços básicos. Estamos comprometidos a não nos silenciarmos frente a qualquer preconceito ou discriminação. Temos que chegar a todos. Com ética e responsabilidade, assumimos a missão de transformar o presente e o futuro, tendo um médico como nossa maior inspiração. E você, o que te inspira na medicina?
Hello everyone, I'm Jaime Ozi, Partner and Business Vice President of the OnixCan Group, the company holding the event. It is a pleasure to have you with us in our event and accept our invitations. Marcos, please, let me go ahead.
Hi everyone, I'm Marcos Bruno. I am editor of the Cannabis and Health portal, the official media outlet for this great event, Medical Cannabis Summit. So first of all, let me introduce our guest, Allyn Howlett. It's a biochemical neuropharmacologist specializing in cannabinoid receptor signal for transduction. She is known in the scientific field for the discovery of the CB1 receptor for cannabinoid agonists, the development of the radioligand binding assay for cannabinoid receptors, and the development of antibodies that were made available to researchers around the world. She is currently a professor in the Department of Physiology and Pharmacology at Wake Forest School of Medicine in Winston-Salem, North Carolina. She is member of the Wake Forest Research Center on Substance Use and Addiction and the P50 Center for Neurobiology of Addiction Management, funded by National Institute of Drug Abuse, the NIDA. Her laboratory investigates cannabinoid and endocannabinoid compounds that stimulate the CB1 receptor and regulate signal transduction pathways in controlled neuronal cells. Dr. Allyn Howlett works with academic and private chemists to study CB1 receptor phosphorylation as a means of targeting signaling through specific G proteins, as well as the potential of drug to target partnerships with different G proteins. The goal is to identify drugs that target neurons in the brain that promote beneficial therapeutics actions. That's it, Dr. Allyn Howlett. It's a great pleasure to be here with you.
Thank you.
Dr. Allyn, first of all, we want to know how you entered in the field of biochemical neuropharmacologist and what motivated you to study the compounds of the cannabis plant.
Well, I'd always had an interest in how the brain worked. And in college, I majored in biochemistry because I thought that would be one of the best ways to study how the brain works. Other people were studying psychology, but I think that working from the point of how the cells interact with each other was better. And I did a very extensive PhD in a pharmacology department, and then a postdoc with Al Gilman, and then went to work in my own lab where I utilized some of the cell cultures that are used for studying how brain cells work. So I've been using these for quite some time, and at the time I was studying how prostaglandins work, and I came across some papers by the Pfizer chemists who were studying how the cannabinoid compounds could be used for pain relief. And at the time, Pfizer was about ready to drop the project as a pain relief project, which in a way is a shame. But on the other hand, the chemists had a lot of compounds. And when Pfizer dropped the project of taking these compounds into the market for pain relief, that let those compounds be available for researchers. So the Pfizer Corporation let me use about 60 of their compounds to do structure-activity relationships. And I think this really helped us move the field forward because that let everyone know that there was probably a receptor involved because the structure-activity relationships paralleled exactly what Pfizer was getting in their pain relief studies in animal models. So Pfizer also helped us along by giving one of the precursors that we could use for making a radioligand binding assay. And they donated that to us. We paid a company to have that radio labeled. And my graduate student, who was William Devane, worked on that project to make a radioligand binding assay. And that was his dissertation project. So that really launched us in the field. We were able to look at signal transduction, how the cells worked, and that paralleled the same pharmacology as Pfizer had for the pain relief.
Very well. It's nice to know that the pharmaceutical like Pfizer previously was studying cannabinoids, and when they dropped the project, they inherited all these information that were very useful for the discoveries that you are about to do, right?
Yes, and I don't think that Pfizer would have published their data if we didn't have our data going along with it. Drug companies don't usually publish everything that they know about the compounds that they have developed. So I think this was a great collaboration between academics and industry.
Thank you.
Dr. Howlett, you are part of this group pioneering research in this area, and you are a great expert in the field of cannabis in particular, now in cannabinoid receptors. So could you tell us more about the discovery of the CB1 receptor, and after the CB2, who was on your team, and how this discovery was important for the science and the movement of the medical cannabis?
Mm-hmm. Up until we started working with the Pfizer compounds and developed the radioligand binding assay, there was great controversy about how cannabinoid compounds work. The reason is that they were very lipid soluble and very much like compounds that work through the, like anesthetics. The general feeling was that perhaps the cannabinoid compounds were getting into the membrane, changing membrane fluidity, and changing the way a number of enzymes that are membrane-bound enzymes would work. And while that's very clearly a possibility, because we knew at the time that there was a great partition coefficient that would make the cannabinoid compounds get into the membranes, much more so than they would stay in the intracellular fluids. So you did have a lot of cannabinoids getting into the membranes. But having a receptor really means that you need stereospecificity, and you need to have a certain pharmacophore that can let those compounds interact with a receptor. And I think that's what our contribution was changing the thinking a little bit. While we certainly know that cannabinoid compounds get into the plasma membrane and they do change membrane fluidity, that what was really happening at very low concentrations was that the receptor was being activated by these compounds. So this led to a pretty good structure-activity relationship profile that we could identify and call that binding site a real receptor. Now, from there were a number of laboratories that had what we call orphan clones. These were 7-transmembrane receptor information from DNA, but they had never been connected to any kind of neurotransmitter or neuromodulator. So they were called orphan clones. And so it wasn't very long after we published our work that we actually had a visitor coming to give a seminar. And when I told him about what we had done, he said, "Oh, I know someone at the NIH who's got an orphan clone that's at the same places in the brain that the cannabinoid receptor is." And when they went back and told them about our poster at the neuroscience meeting, they began to think that their clone was a cannabinoid receptor. And it certainly was. So at that point, Lisa Matsuda was the lead author on that. And that identified the clone that we now call the CB1 receptor. And they were not the only one. There was a group from Europe that also had a clone that was found in the human DNA. And so very similarly, in that same year, both published about this. And this was in the brain. So this was what we now know as CB1. And very shortly after that, the homology was obvious with the CB2 receptor. And again, that was an orphan clone that Sean Monroe had from a cell culture that was a cell culture to study immune responses. So that really identified the CB2 receptor. And at this point, I think it's really unclear about some of the other targets that whether we should actually call them cannabinoid receptors, because their specificity for cannabinoid compounds like THC are not really as great as what might be expected of a true cannabinoid receptor. So I think that brought attention to the field, particularly again from pharmaceutical companies. And clearly other pharmaceutical companies were involved in approaching pain relief from the cannabinoid point of view. But again, those compounds had never really made it to the market.
Okay. Well, doctor, we are talking about the historical to let out the audience to understand how the evolution went. So my question is, the discovery of the endocannabinoid system only happened by the mid of 80s, and the discovery of the two cannabinoid receptors came to be discovered by your team in the early 90s. Is that correct? And almost 30 years after, the chemical structures of CBD and THC were elucidated by Dr. Raphael Mechoulam in the 60s. What do you believe, what owned this delay to? Whether people were not interested in studying the plant, or why it's so difficult to isolate these compounds, which exists in every human being?
I think some of the issues involved here have to do with the difficulty of working with lipid-soluble compounds. One aspect is part of the mindset. At the time, we knew in the 60s that there were neurotransmitters, and of course the sympathetic nervous system and parasympathetic nervous system contributed our knowledge about epinephrine, norepinephrine, and acetylcholine. But back in the 1960s, there were still arguments about whether histamine was a neurotransmitter or even serotonin. And then we came across peptides, and many peptides were found in the brain. And again, there was a question, are these really neurotransmitters? And even in the 1960s, glutamate was not considered a neurotransmitter. There had to be a lot of proof that even that was a neurotransmitter. So now in the 1960s, when Raphael Mechoulam brings up lipid-soluble compounds, I don't think that was so well accepted as being something that could be like a neurotransmitter or a neuromodulator. And of course, today we now know that anandamide and 2-arachidonyl glycerol are very good ligands that interact with the cannabinoid receptor. But in the 1960s, that probably wasn't well accepted. And the second reason is, again, because they're lipid-soluble compounds and they're very prone to sticking to plastic and glass. So people had trouble getting activities because their compounds, if they were electrophysiologists, they had to run through the tubing that would bind up the compounds before they got to be expressed. And then in test tube studies, it was very difficult to get the compounds not to bind to glass. So it was difficult. And when I read the research literature, it seemed to me that one of the best ways to keep the compounds was the way nature does, and that is you carry these compounds in your bloodstream in albumin. So by keeping them in a stock solution like alcohol that could keep them in solution, you could blow dry the alcohol and then take them up in serum albumin. And of course, you need to remove the other compounds that are bound to those lipid binding sites. And so we use fatty acid-free serum albumin. And I think that really helped. So overcoming both the mindset and the structural problems, I think really helped us work with them. And then Billy Martin was working at the same time using animal models. He contributed an assay that could be used in mice or rats that could identify cannabinoid compounds based on 4 different behavioral properties that these compounds had. One was pain relief. Another was a decrease in body temperature. A third was hypomobility in an open field. And the fourth was a measure of mobility that was very much like catalepsy. and could be done either by putting the animal on a ring stand where that's a very abnormal pose for a rodent, and determining how immobile they are. And another way of doing that would be putting them with their front paws on a bar and maintaining that stance over a period of time. So Billy Martin's lab put those 4 assays together in one series that he could use for doing structure-activity relationships that could identify cannabinoid actions in the body. So that was very important, and I think that's really what moved the field forward, was our work with the biochemistry and Billy Martin's work with the animal models.
Yeah.
So, Dr. Allyn, this is a great story. Now we know behind the discovery, the backstage of this discovery. And I ask you, how was the emotion in front of these very important discoveries? Did you know the magnitude of the discovery you were just making?
I think in a very small population of people. So I'll just give you a little example. I had a little girl, a little daughter at the time, and I went to a meeting, and I had taken her with me, and of course she had a babysitter, but one of the people at the meeting said to my little girl, who I think was about 6 years old at the time, she said, "You know, your mommy is famous?" And of course, my little girl's eyes bugged out, and she said, "Oh, really?" Because, of course, the people that I was famous with were the other researchers, not the whole world. But at the time, I was thinking that as we were making progress in understanding the cannabinoid receptor and how it works in neurons and how it works in animal behaviors, with a number of labs that were interested in cannabinoids, it really disappointed me a little bit that researchers who were studying important parts of the brain, like the hippocampus and memory, and the striatal system, and the ventral striatum in addiction, and the cerebellum. There were lots of scientists that are studying these areas of the brain, and yet they were not very interested in what the cannabinoid compounds were doing. And I think that interest really never perked up until the electrophysiologists noted that the endocannabinoid system, the 2-AG and the anandamide, were working as feedback mechanisms. And by that time, we had a number of agonists that were maybe easier to work with than THC. So they were able to identify changes, and they were also able to utilize compounds that could block the synthesis of anandamide and 2-AG in order to demonstrate that neurotransmission could be modulated by these very compounds. So that was a big step forward, but again, that was about 15 years later.
Mm-hmm. Jaime, let me do a question that's not on the script. Dr.
Okay.
Allyn, how is possible? How is possible that human brain and human body have specific receptors for the cannabis? I make a joke: did God create humans and cannabis to interact with each other? And do we also have receptors for other plants, like endo-carrot system, endo-onion system? We have other receptors for other plants?
I think this one is very special because it's a feedback mechanism, and it operates with both glutamate, which is the primary neurotransmitter for excitation, as well as GABA, which is the primary neurotransmitter for inhibition in the brain. And the whole point is that a feedback mechanism really tells the cell that is releasing it and some neighboring cells that they need to revert and go back to what we'd call homeostasis, the way the cells were behaving beforehand. And so in the brain, that means reducing the release of glutamate or the release of GABA so that there isn't as much stimulating the cell that's making the feedback mechanism. So that becomes very important and is a good reason why these receptors are all over the brain. And also they're in many different areas that are involved in what we could consider to be medicinal uses, such things as pain relief. But it explains why we also have side effects like the muddled thinking and the loss of short-term memory, because there are receptors that are going to be in those areas of the brain as well. So it's really a feedback mechanism. It wasn't designed for THC itself. It was designed for anandamide and 2-arachidonoglycerol. And what we know now about other parts of the body is that it's probably a feedback mechanism for those cells as well.
That's great.
Okay.
Brain is
Well,
great.
I think it was from that moment that you began a very proliferating career as a cannabis researcher. And you also played an important role, I understand, in the Pfizer research team, right? However, we know at that time the pharmaceutical didn't show interest in the design studies of pain relief using cannabinoids and judged cannabinoids as an incapable compound to be administered for pain relief and home use. Could you tell us about this experience in the eyes of the company at that time, and how your studies has influenced the change of the opinion of this company?
Well, I think we have to realize that because these receptors are all over the brain and in many different areas, that targeting these receptors means that we also have side effects. And I think that drug companies, and particularly at about that era, were really a little concerned about what side effects would be for a drug that would be expected to be home use, as you said. If you're going to have chronic pain and you're going to be using something for pain relief, you have to deal with how the side effects work. And people who were using aspirin-like compounds were having to deal with side effects such as internal bleeding and intestinal problems, because those were other systems of the body that were responding to the nonsteroidal anti-inflammatory agents. On the other hand, people who were using morphine-like compounds had to deal with the addiction and the sedation and the possibility of overdoses. So on the one hand, developing cannabinoids would have very different side effects from either of those two uses. But you can understand how there might be a liability of sedation and the loss of short-term memory and the muddled thinking, or what scientists would call cognitive dysfunction, with using cannabinoids at the levels that you would need possibly for pain relief. And at that point, we didn't really know very much about CB2 receptors, and we didn't have selectivity, because the compounds that we knew worked with CB1 receptors also worked with CB2 receptors. We're much further along now because we can appreciate neuropathic pain and we can appreciate inflammatory pain as being something that can be influenced by the CB2 receptors, and maybe even cannabidiol, at a much better way than the CB1 receptors. So we now have a large number of CB2 receptor analogs, and we have an interest now in how CBD works. And I think we're going to be moving much more quickly towards what could be useful pain relievers based on those aspects of cannabinoid receptors.
Okay, Dr. Howlett, we would like to hear from you your true opinion on the use of synthetic cannabis compounds such as THC. Why are pharmaceutical companies interested in these types of compounds over natural plants compounds? Is it a commercial strategy for popular acceptance? Is it to circumvent the laws or to develop patented and more potent drugs?
Well, we certainly had more potent compounds. The Pfizer compounds were very potent, and they were not the only company back in the 1980s that were working on cannabinoids as pain relievers. Abbott and Lilly also had compounds that were very similar to THC. And the reason that drug companies make modifications on plant products is for many reasons. One is to keep it in the body a little bit longer. Another would be to have some selectivity for how that compound works. And I think back in the 1980s, we were thinking that there were probably many more receptors than just CB1 and CB2. And in looking for other receptors, we couldn't find anything that would be more selective. You know, you would want to have a receptor that was in the pathway for pain relief, but not in the pathways for, let's say, the hippocampus for memory consolidation. And we were never able to separate side effects from the beneficial effects until we knew a little bit more about the endocannabinoid system. So I think that was a major impediment for CB1. In some of the studies, and particularly those from John Hoffman, it was fairly obvious that working with some of the Stirling-Winthrop compounds, these were based on an indole nucleus, that John Hoffman was able to separate beneficial effects from some of the side effects by looking at CB2 receptors. And he found a number of compounds that would work through CB2 receptors, but not CB1. And then drug companies became interested in this as anti-inflammatory or neuromodulators for pain relief based on CB2. So we've had some changes in the way we were thinking, but that all has to do with selectivity. And that could separate CB1 and CB2, but those are the only two receptors that we have. So if we're going to be looking at things that are going on in the brain, we have to worry a lot about those side effects because it's the same receptor, whether it's in areas of the brain that are involved in metabolism and eating behaviors, or whether it's in part of the brain that has to do with pain relief. Either one of these could be targets, but we still have to worry about the side effects. And we have to worry about the side effects that come from antagonists as well. So this really has been the major impediment, is separating the side effects and causing selectivity. And that doesn't necessarily happen if you take pure THC from the plant. But now I think we appreciate that some of the other compounds that are in the plant probably have beneficial effects that haven't been looked at. And right in Brazil was where it was noted, and there was clinical trials on CBD and epilepsy back in the early 1980s. Unfortunately, drug companies didn't really pay much attention to that. And I always wondered why, because in that period of time, the only two drugs we had for treating seizures were phenytoin and phenobarbital, and they have very difficult side effects. But CBD didn't appear to have those same side effects. So I'm glad that we've had a resurgence in interest in CBD, particularly with the involvement of seizure control.
Okay, so probably you're talking about Dr. Elisaldo Carlini that started those
That's
researchers
right.
here in Brazil. Yeah,
That's
he did a
right.
wonderful job, no doubts about it. Well,
That's right.
can you, it has been so interesting to understand. I just want to have a brief summary from you how cannabis works in the brain. If you're not let's say a scientist or researcher, in a simple way, how it works, why it has these ligands, how it works with the neuroreceptors. Can you just give us a brief idea how it works in the brain?
Yes, it's part of a feedback mechanism. So when you have neurons in the brain, these are the brain cells that communicate with each other, and they communicate all over the brain. So we have the release of neurotransmitters that were packaged in what we call vesicles, and they're stored, and they wait for an electrical signal that can come to the end of what we call an axon, and that releases those neurotransmitters onto the next cell in line. So it would be similar to communicating that you would have with people in your neighborhood. You might put a letter in the mail, and then the letter gets delivered to the door of your neighbors. So once that release of vesicles is made, you could have an overstimulation. So you need to have a feedback mechanism that says, yes, we've got plenty of neurotransmitter receptors being stimulated. We don't need to make any more neurotransmitter and release it. So that mechanism is what the endocannabinoid system does. Endo means endogenous. That means the way it works naturally in the body. And the endocannabinoid system is based not on THC, but on two compounds that the cell makes. One is called 2-arachidonoglycerol, and the other one is called anandamide. And these are compounds that are made when the cell gets overstimulated. And they're, again, very lipid soluble, as is THC. So they can cross the synapse to get to the side that has the release properties and is going to be releasing the compounds in the vesicles, and it will block that release. So it really helps as a feedback to shut down the production and release of new neurotransmitters for a certain period of time until things can get back to normal. And once they get back to normal, then that system really isn't needed anymore. So it's a feedback mechanism.
Very well, thank you. Very clear now.
The brain is amazing. It's a grateful gift from God. Dr. Allyn, we are now going to reproduce a question from our medical partner, Dr. Ana Gabriela Hounie. She is a psychiatrist and a pediatrician and one of the pioneers in researching and prescribing cannabis here in Brazil. And her question is about neurogenesis and autism.
Dear Dr. Howlett, there is evidence that CBD and some other cannabinoids, such as CBG and THC, have neurogenic properties. Considering the benefit of CBD in autistic patients, what do you think is the role of neurogenesis in the improvement of their symptoms?
I think this is an area that needs a lot more research and attention. And I think if you begin to think about treatment, you have to go back to potential causes. With autism, we have a problem because we really don't know what the potential cause is, but we can imagine that it probably has to do with some development in the brain. And we know now that the endocannabinoid system is very important in making neuron connections. And by that, I mean, when the neurons start from stem cells, they move to neuroprogenitor cells. And as these neuroprogenitor cells receive information from the other cells in their neighborhood, they begin to determine whether they are going to be neurons, glia, or oligodendrocytes. And once they've made that commitment, the neurons will no longer divide, but they will start to migrate, and they'll go to places where they need to be, and they'll send out projections that send axons or dendrites to where they need to go. And if that problem is disturbed anywhere along the way in development, then there can be problems that have to be compensated for by the rest of the brain. And I think we probably realize that the brain will make as many compensations as it can, but maybe there are some compensations that just can't be made. So I think we need to think in terms of how these things are being modulated by the endocannabinoid system and whether autism might have an involvement with endocannabinoid responses during development. Now, your question had to do with after the baby is born and we realize that there must have been a problem in the development and we have autism, how can we deal with that? And I think there are many things that are related to behaviors in people that maybe can be harnessed. I don't know how we're going to treat autism, but we don't know very much about how cannabidiol works. So this is an area, again, that needs a great deal more research. We have some ideas that cannabidiol may inhibit some of the effects of THC in neurons in the brain, but I'm pretty sure it must have other effects as well that we don't really have good understanding of. So I think what we need to do is have much more research on brain development, on feedback mechanisms in the brain, areas in the brain that are responding in autism, and maybe some effects that we can understand more about how CBD works.
Well, Doctor, you have been witness the debates and discussions over the U.S. federal reports for the encouragement of cannabis research. In your opinion, in the recent years, how federal legislation affected the field of cannabis research?
Federal regulations, I think, that started back in the 1930s and 1940s in the United States really curtailed the use of cannabis, but those were purposes that were related to recreational use, not necessarily to medicinal use. And at the time, I think physicians really didn't realize what the properties of cannabis were and really didn't care. So the medical profession was not concerned about whether we had cannabis available or whether we didn't. So that went on, and I think now we have a great deal more understanding about THC, what its properties are, how it works in the brain, what we can use it for. But, you know, as we've discussed, there's not good selectivity with THC. Because of the extreme interest in THC and the receptor and the many ways in which the cells in the body work, we pretty much ignored CBD for a number of years, and we ignored other kinds of plant products. So it's just recently that we've been able to work with those, and those have been under the umbrella of the federal regulations that call things cannabis, even though that means that these compounds were not having psychoactive effects on brain function, but we just don't know very much about what their potential are or how they work. So we've now got a new era in which we're able to study cannabinoids in many different ways, and we understand how the cannabinoid THC works in many different areas of the body and in the brain. And I think we've got tools now as molecular biologists to understand how some of these other compounds work as well. So we're all set to begin to look at how CBD and some of the other compounds that are found in the plant product might be useful in treating certain kinds of diseases. The impediments now are the protections that we as consumers have, not just here in the United States with our Food and Drug Administration, but with the European drug organizations and throughout the rest of the world. There are really regulations that keep drug companies so that safe and effective medicines are available for consumers. And those have to do with regulations about, as we just said, does it work? Does it work with the way people in the drug companies say it works? And that means not only do you need to have animal models, but you also need to have clinical trials. And the other aspect is it safe? And so again, you need animal models, you need to have biochemical studies, and you need to have safety demonstrated in clinical trials. So this is now a situation that consumers who are going to be looking at plant products need to consider just as much as consumers of medicines that are made by chemists in research laboratories or in the pharmaceutical industry. So I think we have to adjust our thinking now to treat these compounds in much the same way as the pharmaceutical companies treat every other compound that they make into a medicine. It has to have safety demonstrated, and it has to have a demonstration that it works.
Okay. One more thing regarding this. If cannabis is not totally legal as far as federal legislation is concerned, do you have any difficulty in raising funds or even in the, let's say, the ethical commissions to pass those studies to have more, let's say, openness in order to make this research and make this research fruitful?
Well, in the United States, most researchers that work in diseases are using funds from the National Institute on Drug Abuse. And that's where most of my work has been done. There are research in many different areas, whether it has to do with addiction or simply how cannabis works, has been funded by the National Institute on Drug Abuse. Not necessarily the idea that the National Institute on Drug Abuse is going to provide people with different kinds of plants, although they do that. Many of us use the National Institute on Drug Abuse sources for some of the compounds that we use. However, the National Institute on Drug Abuse has been very, very helpful in supporting research going forward. Now that we know that the endocannabinoid system is involved in other things such as cancer and metabolism, bone growth, many, many areas of the body, liver dysfunction, it's time that some of the grants that people submit to get funding should be considered from some of these other disease states. And right now, I think that hasn't really been something that other institutes at the NIH have considered to be as important. I think we need to have a little mindset change there. In the rest of the world, there are many, many laboratories studying cannabis. And these laboratories are getting their funding from other kinds of sources, but the researchers throughout the world, let me give you an example of the membership of the International Cannabinoid Research Society is greater from the Israeli and Spain and Great Britain markets than in the United States. But, you know, throughout the rest of the world, we're all getting our funding from either national or foundation sources. And I think, you know, we're competing with other people, other laboratories that are studying other kinds of things. So we're all competing for the same pot of money. It's not that there's any discrimination necessarily for people studying cannabis.
Okay, well, that's great. How taking this this opportunity of what you said, what can we expect in terms of new treatments that will come after this all this investment or all these researchers involved in the research of cannabis? What can we expect?
Well, I think one area that has been uncovered that is going to be groundbreaking is metabolism. And that's because not only in United States, but throughout the world, there are a good deal of people with diabetes and cardiovascular disease. And the treatments may be very different, but we do have the opportunity to think in terms of the cannabinoid system being part of a pathway that can reward eating of dietary compounds that are high energy compounds. That is, they have a lot of fat and sugars in them, and potentially blocking cannabinoid responses, maybe not in the brain, where again there are side effects, but in the areas that are involved in metabolism, such as the liver and the pancreas and skeletal muscle. All of these areas, I think, are just ripe for some modifications that can perhaps help us with this obesity and metabolism problem throughout the world. That's one area. We just had a question about autism, and I think we can also begin to think about schizophrenia. These are areas that are involving the brain that we don't really have a good understanding about what the original cause was, much less how we can begin to treat it after development has been perturbed. So these are areas that are what we have unmet needs for diagnostics as well as treatments. And perhaps the cannabinoid system can be involved there.
Dr. Allyn, here in Brazil, less than 1% of medical professionals prescribe cannabis to their patients. The endocannabinoid system is not even taught in the university courses, both in the health area and especially for doctors. So I ask you, is the endocannabinoid system taught at all the universities around the world? What do you think about this problem? Because it's been 3 decades since the discovery of endocannabinoid system, and it's not taught in university.
Isn't that something? Because I think if you're going to start at a younger age, it would be in the cell biology courses that you would really want to begin with these receptors and think in terms of them as a feedback mechanism. And you're right, there aren't any signal transduction pathways that specify cannabinoid receptors in early pre-med kinds of courses. And then the curriculum is very, very heavy in MD training. And I think that at this point, if we don't have a drug, then the pharmacology is not going to be taught. So the cannabis section of teaching in medical school tends to be related to substance abuse. And that's where cannabis gets included when you talk about cocaine and crack cocaine and amphetamines and even ethanol. And it makes it look as if cannabis is a very evil drug, but it doesn't really present opportunities because at this point, we're just beginning to get medicines. So I think that's going to be changing because more and more throughout the world, GW Pharmaceuticals has inroads and approvals for use of two compounds, either Sativex or Epidiolex for seizures. And it's there that I think the physicians are going to begin to get their medical education, because now we have a drug that we can point to. The use of THC for AIDS and AIDS wasting, that's not an issue anymore because people who have HIV are probably being treated with antiviral drugs. And so that's not necessarily an issue.
Well, doctor, how do you see the future of cannabis, let's say 20 years from now, looking more ahead?
Well, I think that many of the products that we have on the market currently are originated from plant products. And so with THC, that's really not very much of a difference, is it? But when we think about cannabis compounds themselves, I think we have to look at some of the other compounds that are in the plant. We already know that THC is not very selective between CB1 and CB2 or any parts of the brain. And so there, the pharmaceutical industry has to really deal with selectivity and try to determine what is unique about some of the areas of the brain that are going to be involved in therapeutic uses, such as pain control or metabolism regulation, and develop some selectivity for agonists and antagonists that can direct that. So there's where we need academic assistance, because it's in the academic research fields that people begin looking for new classes of compounds that are going to be analogs and can really move the field forward in that direction.
I hope the medicine will come back to the ancestrality, to the plants, to the mushrooms, and the things that the nature gives us. So, Dr. Allyn, it was a great review. I'm very proud, me and Jaime. I know Jaime is proud too, of being here with you, told all your knowledge for us. For me, professionally, it's very proud to be here with you. But we have just a few minutes, and I have to make one last question for you. And I ask you, what message would you like to leave to the doctors and patients who assist us now, especially those who are looking for more knowledge about cannabinoid medicine?
Well, we've got lots of knowledge available in the research literature because we've got a number of people throughout the world who are studying cannabinoid compounds, and they write review articles which are available for those who are younger researchers that want to move into the field. We also have a number of research groups. We have the International Cannabinoid Research Society, and then we have lots of smaller groups, a European group, and areas that are Gordon conferences throughout the rest of the world. So these are places where scientists can meet and talk to each other and expand their knowledge. What we have to worry about, I think, a little bit is the media, social media, in which information may be disseminated that may not be quite true or may not be highly investigated. So finding sources from social media that are going to provide accurate information on cannabinoid drugs and cannabinoid drug use, I think we have to, maybe as research scientists, have to do a better job of communicating that kind of interest to the patient population. And as you pointed out, medical schools are not necessarily teaching about the endocannabinoid system because there haven't been very many drugs that have come from that are used now for treatments. However, I think that's another area where researchers can begin to provide that kind of information right in our own medical schools so that we can inform the future physicians and the current physicians about how this feedback mechanism works in the brain and where cannabinoid receptors are in the rest of the body and how it can relate to the kinds of medicine that other people are using.
I think that's it. Jaime, can you ask
Yes.
Dr. Allyn the last words and
Dr. Allyn,
ending our interview?
it couldn't be better. I wish we could have more time and explore so many ways that you open up for the scientists, for the patients, because it's a different situation from all the medical developments. We see here in Brazil and probably everywhere, the demand of these products came from the patients that they don't have, as you said, their needs attended. So what we see here, even having just 1% of the doctors prescribing, and this is why we are investing in qualified information like the interview that we are doing with you, is to provide for not only for doctors, not only for physicians, but for the whole population, qualified information where they can have their decisions. So thank you very much. You are giving us the opportunity to abroad, to enlarge the knowledge about cannabis as a whole. Thank you very much. We look forward to see you again in our meetings. Thank you.
Mm-hmm. Thank you. Bye-bye.